So hi everyone, and welcome to our October Carriganine Science Series webinar. My name is Amanda Akis. For those of you that don't know me, I'm a fish biologist at the USGS Great Lakes Science Center in Ann Arbor, Michigan. And today I'm going to be moderating with Nick Boucher from the Great Lakes Fishery Commission. And today marks a series of 3 fantastic seminars on whitefishes that we have lined up to finish out the year. Just to highlight what we have coming up after this seminar, in November, we'll be hosting a seminar by Dr. Andrew Hunze and future Dr. Taylor Brown on lake whitefish recruitment. And in December, we'll be hosting Dr. Louis Bernichet for an overview of 30 years of lake whitefish research from his lab. So we hope that you will also be able to join us for those seminars. And just a little reminder and plug that if any of you all have relevant Carriganine research that you would like us to highlight in one of these upcoming seminars for 2023, don't hesitate to reach out to us and let us know that you're interested and have something you'd like to share. So today, it is my pleasure to introduce our speaker that starts this series of whitefish talks, Dr. Philine Feulner. Dr. Feulner is a group leader in fish genomics at EAWAG, the Swiss Federal Institute of Aquatic Science and Technology. Her research focuses on understanding how evolutionary processes shape genome evolution, and we're very excited to have her here today to discuss a genomic perspective on speciation and adaptation in alpine whitefish. We will have some time for questions and discussion afterwards. So if any questions arise during the presentation, feel free to find the chat and type those in the chat and we can highlight those. Or you can hang on to those questions and raise your hand afterwards and ask them in person. Welcome, Dr. Feulner, and thank you so much for joining us today.
Thanks so much, Amanda, for the invitation. I'm really thrilled to be here. I will now try to share the screen and the presentation. Is this working all right?
Yep, looks great.
Yep. Okay, then yes, so thanks so much for that invitation. I already said that I'm really excited. I think I'm mainly spoken in front of European audiences, so I'm like really pleased that I get the chance to present the work also to a wider audience today. And thanks so much, Amanda, for making that possible, and Nick for organizing all of this. And yeah, I want to talk about the whitefish we actually have here in the alpine lakes just outside of the window. And I work at EAWAG, which is, like Amanda said already, the Swiss Institute for Aquatic Technology and Sciences here in Switzerland. And some of my colleagues are actually like building really technology for the aquatic sectors, while other colleagues, including me, are concerned about the aquatic ecosystem and the biodiversity in aquatic ecosystems. And we are like all, and I guess you guys as well, are like seeing these dramatic changes of ecosystems, and often really due to human actions. And we see that in the terrestrial environment, but we also see that in the aquatic environment. And due to these dramatic changes, we also observe this loss of biodiversity in these environments. And it has been shown that especially in the aquatic and in the freshwater habitat, we are losing even more alarming, or like these losses of biodiversity are even more alarming than in other ecosystems. It's also interesting, at least for me, from like I'm an evolutionary biologist by training, to see that many organisms in the freshwater actually evolve due to the heterogeneity of freshwater habitats. So they evolve due to a process which we call ecological speciation, where the variation in different environments creates like different fitness peaks, and then divergent natural selection can actually drive the differentiation between species. And this is a process where reproductive isolation is to a large extent relying on stable ecological conditions. And when I think about ecological speciation, I think about this like as a continuous process where over time, this divergent natural selection builds up reproductive isolation between species, and they're like diverging over time. And I think me and also others have like thought about this, that the evolution of intrinsic environmental independent reproductive isolation mechanisms are taking a long time, while this process can be quite fast. So really, like in many cases, there has been no evolution of intrinsic environmental independent mechanisms for reproductive isolation. And that has also been demonstrated by the fact that this process can also be reversed when actually environmental conditions are changing. And there have been a few examples where this happened, where like when, for example, lakes got, or like water got turbid, like reproductive isolation broken down again, and the differentiation between species got declining again instead of like growing. And in my lab, I'm interested in like observing these processes and also observing the biodiversity in this alpine whitefish radiation, which you already see here at first glimpse, but then also by being invited by people from the Great Lakes, I, for myself, and maybe it's interesting for you as well, had to bring this into like a perspective of sizes. And it was quite revealing for me to see that actually the whole of Switzerland, so everything I'm concerned with in my research, is at a size like somewhere in between Lake Erie and Lake Michigan. So of course, even if I think of these lakes as being big, they're like orders of magnitude smaller than the Great Lakes you guys are studying. And actually the two largest lakes in Switzerland are Lake Geneva and Lake Constance, and they're really like 2 orders of magnitude smaller than the Great Lakes. But they are also fairly deep lakes. So they are like a few hundred meters deep. And naturally, these are like oligotrophic lakes. They're really nice and clear. And they host like this amazing diversity of whitefish. We have many lakes with at least 2 species, so like 6 lakes which have 2 species or more. We actually have 2 lakes for which now 6 different species have been taxonomically described. One of them is Lake Luzern, that's where our institute is, and another one is Lake Thun. It has been demonstrated in the past based on small number of markers that this alpine whitefish, which are distributed in peri-alpine lakes, they also occur like in some peri-alpine lakes in Austria and southern Germany, that they form a monophyletic group within the European whitefish lineage, which is the Coregonus laberatus, so the sister lineage to the Coregonus crupiiformis, which are like distributed in Northern America. Previous work also has been indicating that actually individual species which live in the same lakes are each other's closest relatives. So that these are really like sort of mini radiations occurring in having happened in these lake systems. But across different lakes, we often find similar ecomorphs. But these similar ecomorphs are not each other's closest relatives. These lakes also, especially due to the proximity of humans to these lakes here in Europe, have been heavily affected by something which we call cultural eutrophication. So to the middle, to the end of the last century, there has been an increased intake of phosphate into these lakes. And you can see that here. These are like phosphate curves from multiple of the Swiss lakes. It's actually a transient environmental disturbance. So like after the implementation of wastewater treatment plants, this could be reverted again. And nowadays, these lakes are still, again, like oligotrophic, nice, clear lakes. But during this time, when there was this increased intake of nutrients, oxygen levels, especially in the deep areas of the lakes, have declined dramatically. And there has been a shift in the ecosystem where like more food abundance was happening in the pelagic area of the lakes. And all of this has also heavily affected the whitefish. And we lost many historically described whitefish species all across Switzerland. And in this paper from 2012, which is also done by colleagues here at EAWAG, they could demonstrate that there has been a correlation between how much a lake was affected by this phosphate intake and how many species we lost. But all this past work was actually done on like microsatellites, mitochondrial data, a few AFLP loci. But when I came to Switzerland, I really wanted to study this system from a genomics perspective. And what I want to understand in my lab is like how ecological factors and evolutionary processes influence the genomes of organisms. And I want to I look at the patterns of variation and differentiation in the genome and try to infer which processes shape these genomes. But I'm also interested to understand which properties of the genome actually allow ecological or evolutionary processes, like what allows the persistence of different species with each other, or what's constraining or facilitating adaptation and speciation. But to be able to do this in this whitefish system, the first thing we really had to do was to assemble a reference genome. And I think I saw him and it made me a bit nervous, but my PhD student, my former PhD student, Rishi Decane, might be in the audience. And he really led this part of the work. I guess it's in this audience, it's obvious that corygonids are part of salmonids, and they have fairly large genomes, and they have a fairly recent whole genome duplication. So they are a challenging organism to assemble a genome for. Nevertheless, Rishi together with a bioinformatician from the Genomic Diversity Center in Zurich, took up this challenge and established this pipeline, testing 3 different assembling methods to actually compile this reference genome. And he also had, before that, assembled a linkage map. So we could, at the end, compare the linkage map, which is on this side here, with the genome assembly we gained. And these lines show you, when they're going across very nicely, that there has been a good synteny between the linkage group and the reference genome. And you can see, overall, I think it looks quite nice, but there are these regions where things go in different direction, where we have poor synteny between these 2 methods. And interestingly, Rishi also highlighted all these regions where we have these mismatches in gray. These are all regions of the genome when we map short read data against the reference genome. We have an increase in coverage and an increase in variation of coverage. And that's pretty good indication that these might be these challenging regions of the whitefish genome where things are still highly sequence similar and duplicated. And during the assembly process, two similar regions actually have been collapsed into one region. And in our case, this is about 14% of the reference genome. It sounds like a big number, but I'm actually fairly pleased with it because the Atlantic salmon reference genome still has 9% of this unresolved regions. So we were pretty happy about our genome assembly, but both of us, Rishi and me, are actually really way more interested in understanding evolutionary processes than solving bioinformatics puzzles. And this is also what I want to spend the most time of my talk today. Is one study where we looked at the genomic variation across a spatial scale across different lakes in Switzerland. And one study where for this lake, Lake Constance, which is the 2nd largest lake in Switzerland, we also have samples from historical scale collections. So we can actually look at genomic variation across the temporal scale. But I want to start with the spatial scale, which is really the last chapter of Rishi's PhD thesis, where he looked at a total of 99 whole genomes, which we had, and where we sampled all the lakes in Switzerland where there's still like 2 species which are natural species occurring in these lakes. And they are like from 5 lake systems. So it's a total of 8 lakes, but some of these lakes are like connected or have historically been actually one lake. So we call these like larger lake systems. And the different colors are like reflecting these 5 different lake systems. And in this lakes, we find a total of 22 different taxonomically described species. But there are like basically 6 ecomorphs, which we can find. So we have like an ecomorph differentiation across depths, where we have a larger species, which in Switzerland we call like a Balchen type, which is a large benthic shallow spawning species. And then like we have something like deeper down where the species become a bit smaller and also become a bit more pelagic, which you can also see in their like gill raker morphology. So they get more and finer gill rakers. In some lakes, we have a morph which really lives in the pelagic waters. And also in Lake Constance, it actually is an ecomorph which spawns pelagically. And in some of the lakes, we have something like profundal ecomorphs, which live and spawn in the really deep areas of the lakes. And when Rishi uses used this whole genome data and created a phylogeny, we could see a few things which like are consistent with previous phylogenetic studies on way fewer data. So we see that again, all this like alpine whitefish form a monophyletic clade. And we, in this study, really only have very few outgroups, but we have outgroups from Scandinavia and they are like down here. And you can also see that the species cluster very nicely by color. So that actually really like fish from one lake, which are the same color, are each other's closest relative. And they don't cluster by their ecomorph symbol. So it's not like that the different ecomorphs are each other's closest relatives. It's really like fish within the lakes, which are each other's closest relatives. And I don't know, because there's lots going on in this slide. You also see that there seems to be some individuals which look a bit mixed. And maybe you can also spot that like we have this one case where like it's actually not what I just said, like that all the fish from the same lake are in one cluster. And this is actually concerning one of these pelagic morphs I talked about, where like we have this large pelagic feeding ecomorph. And in this case, we actually know there are historical records that pelagic fish from Lake Constance have been introduced by humans and stocked by humans into Lake Thun. So like while these fish occur in Lake Thun, they are like the relatives of fish which were brought from Lake Constance into Lake Thun. And by seeing this, like Rishi also explored that a bit more, like looking for like where along the tree we find signatures that the tree is actually not the best explanation for the data and the relationships between these fish. And he used these statistics, which give you like an indication of an excess allele sharing and like is like an evidence for introgression. And all these events where there's a significant evidence for introgression are like in these dots in this figure, and how much of this introgression has happened is like indicated by how deeply red a square is formed. And it's like sort of a complicated figure, and it's like you see that like there are multiple of these events, but by highlighting these ecomorphs where we have this large pelagic ecomorphs, you can see that these are often evolved in these introgression events. And these are like here in this like squares. So what we think is the best explanation for this one ecomorph is that seems to have only evolved once in Lake Constance, and then like was likely by humans introduced into both Lake Thun and Lake Lucerne. So it's like sort of a secondary contact event in these lakes. And we see introgression between the introduced fish and the native fish. But also nowadays, really, these ecomorphs like formed like a species, and they are genetically distinct species which persist within these natural radiations in these lakes. So sometimes the secondary contact and this human-mediated transfers seem to have like provided some of this diversity. But we also find repeatedly this differentiation along depth in these lakes. And these events are usually not correlated with any introgression pattern. So it really seems like that this is like something which has repeatedly evolved and very rapidly evolved. So we were wondering a bit like, can we see something in the genetic architecture of these ecomorphs, potentially explaining why this could have happened so rapidly? And I'm sure also everybody here is very familiar with like the gill rakers, which sit on top of the gills and help this fish to filter feed the organisms they live on. And there is this gradient in whitefish where like the more benthic species have fewer gill rakers, and they're like usually shorter. And the more pelagic feeding whitefish have many gill rakers. And actually, Rishi just set out to like find an association of the gill raker count with the genetic data. And here you can see like one of these GWAS plots, and that's for gill raker count. And you see that there's very little along the genome, but we find this one place where like we just about hit the significant threshold. And it's interesting because at that region of the genome, we find a gene annotated, which is called the EAWAG gene. And that gene is known for being involved in the development of ectodermal tissues. And actually, if this gene gets knocked out in zebrafish, that affects the gill rakers of zebrafish. So it's a pretty good candidate gene for like variation in gill raker count. Based on this analysis, we can approximate that about like 30% of the phenotypic variation we see in our data set is explained by this one locus. But 30% is really like not everything. So Rishi also set out to try other methods to understand the differences between these different ecomorphs. And one thing we could do is we could contrast two fairly extreme ecomorphs, which are very different in their gill raker count and also are different in their size, which have repeatedly evolved in 4 of these different lake systems. And we can look like, where is this differentiation happening in parallel along the genome? And this is the plot you can see here, and all like the significant outlier loci are now in this dark black dots. And you can see that it includes this peak we have identified as a GWAS peak for gill raker count. But you can also see that many loci all over the genome are also involved. And genetic differentiation only at those loci is actually correlated with gill raker count and negatively correlated with the size of the species. So it's not only like this one major locus, it's also many loci all over the genome which are evolved in this underlying this ecomorph differentiation, and potentially, especially this architecture as a mix of a major effect loci, many loci could have contributed to the rapid and repeated differentiation. So this is already one of my summary and take-home messages, which I just said, that we think that the rapid and repeated divergence of ecomorphs in multiple lake systems has been facilitated by this, what some people have termed mixed genetic architecture. And we also seen this evidence for introgression upon secondary contact, but that resulted in the persistence of actually pelagic ecomorphs within natural radiations in these lakes. So this is summing up the part on the spatial variation. And now I want to move into this study of one of the lakes, Lake Constance, where there are like 4 described whitefish species. We have one of these pelagic morphs. We have this large benthic morph and a bit smaller, more pelagic morph. And also in this lake, there used to be one of these profundal ecomorphs, which actually got extinct. And another really talented PhD student of mine, David Fry, he grew up on that lake, is actually fishing this fish for a long time of his life, and was really excited to work on that. And he was also brave enough to take up the challenge to actually work with DNA from scale samples. Because for this system, we have this historical scale collection, which actually was sampled before the onset of the eutrophication. And we also have scale samples for the species which nowadays is extinct. And these samples have been previously used for microsatellite work. And there has been already some indication that some microsatellite alleles have been introgressed from the extinct species into the contemporary species. But nobody has looked at that from a genomic perspective. And David was really good in extracting this DNA, and we actually were managing to sequence whole genomes from these scale samples. And in his plots, he actually uses this crosses for the scale samples, and the dots are like used for the samples which have been sampled after eutrophication. And this is like one of his first analysis, which is just a PCA plot where you can see the relationship of all the samples to each other. And what you can see is that the extinct species seem to have been fairly distinct from the other species. And you can also see that all the before eutrophication samples seem to be more differentiated from each other. And the more contemporary samples are more similar to each other. So they're moving into the middle of this PCA plot. If we look at that in like a mixture proportion plot, what you can see here is, so he's using gray for the extinct samples, that in all the samples which we looked at, which we sampled before eutrophication, we don't find evidence for admixture from the extinct species. But only in the contemporary samples, we find these gray bits. So again, like confirming what we're seeing from microsatellite work. But the setup with like having extinct species is now very similar to the setup of the people having demonstrated the introgression from Neanderthals into modern humans, because we have the past samples and the contemporary samples. And so you can use exactly that setup and the D statistics, which was used to demonstrate introgressions from Neanderthals, and used it for demonstrating that exactly that's happening also in this whitefish system, where we see significant introgression, also access allele sharing between the extinct species and the contemporary species. And when he looked at the whole radiation, he actually could see that there has been this introgression from the extinct species into all 3 species which we find nowadays in the lake. But then that we can only see introgression events into specific directions. And it's interesting because the introgression seems to progress exactly the same way how the habitat collapsed during eutrophication. So it's from the deep where the oxygen was depleted into the more shallow areas, and it's from the benthic towards the pelagic habitat. So really following the breakdown of the habitat. But we have now this whole genome data, so we don't only want to like use like it to summarize it across all this massive amount of information. We can also look at where in the genome do we find evidence for introgression. And for that, David used like a topology weighting approach, TWIST, which was developed by Simon Martin in Edinburgh. And with that, he identified the regions of the genome where we find introgression. And he like has these 3 different colors for the 3 different species, and the gray tick marks, which I here like highlighted chromosome 10, these are the regions of the genome which show evidence for introgression. And you can't see that very well on this circular plot, but really like you can trust me that all the chromosomes look a bit like this one here. So it's really like widely distributed across the whole genome. When he sums it up, it looks like about 28% across all these individuals we sequenced are still like variation from the extinct species having introgressed into the surviving species. When he looks like which individuals share specific regions of introgression amongst each other, it's interesting to see that there's more introgression shared between individuals of the same species than between 2 individuals from different species. So like while there has been all this mixing, there's still like a differentiation between the species, and the species still stay distinct besides having received all this gene flow. We can also look like to which respect do these introgressed regions overlap with genes and exons. And here you always see the introgressed versus the non-introgressed regions, and this is the overlap with genes, and this is the overlap with exons. And as you can see, there's no difference. And I think that's pretty interesting, because my expectation would have been that maybe there's like some selection against introgression in genic regions. But so this seems to be not going on here. So no purifying selection against this introgression. And rather on the contrary, like another thing, because we have 11 genomes from the extinct species, we set out to run a scan for selection on this extinct species to identify the regions of the genome which could have been under selection in the extinct species. And then we can ask the question, to which respect do those regions overlap with the regions which have been introgressed into the other species? And we found this huge overlap, like it's 50% of the selected regions which also seem to have introgressed into the other species. And that's pretty interesting, like just as an observation, because nowadays, as I said before, like wastewater treatment plants have really changed the ecosystems again. And this habitat, which used to be occupied by the extinct species, is now available again. And there's oxygen in these deep areas again, but there are no whitefish there. So David also set out to put nets in a depth gradient and catch whitefish on an old spawning ground, which we know as a spawning ground of the extinct species. And this species is a species which we know has a broad spawning gradient, and it's ecologically potentially the closest to the extinct species. Nevertheless, like in the literature, like it's reported that it spawns down to something like 40 or 50 meters. But also to the surprise of the fisher wardens at Lake Constance, David actually could catch fish also down to 90 meters. And if he looks at, again, doing whole genome data, how these fish like are differentiated, it's interesting. They are all like one species, and like, yes, there's not much variation explained by this PCA axis, but there is this slight differentiation we can see in the 90-meter fish. So those fish which really, really like have been during spawning seasons been caught at a very deep net. Okay, the first thing I wanted him to check is like, do actually maybe these fish which we caught so deep have more introgression? But there's not much variation. So there's like variation between the individuals, but like no consistent variation of like an increase of the amount of introgression of any of these nets. But when he looked at like which regions in the genome are like differentiated between the 90-meter fish and the other fish, he could identify a few locations along the genome. Again, spread out on different chromosomes, multiple loci, With this cutoff, we have like 52, I think, outlier loci. And some of them actually show a pattern like the one down here, where like it's not surprising, like they have like a different allele frequency in the deeper nets, and they have an increase of one allele in the 90-meter fish. That's exactly the pattern we looked for. But then if we look at like what has been the allele frequency in the extinct species, it looks like that the allele frequency of the 90-meter fish more matches the allele frequency of the extinct fish. So we feel like that's like at least a pattern which could be consistent with adaptive introgression from the extinct into the Coregonus macrophthalmus species here. And this is the pattern which he sees across all these 52 loci. And actually 12 of these 52 loci show a pattern which would be consistent with adaptive introgression from the extinct into the modern contemporary species. And if it looks like, how does that compare to like random loci all over the genome? This seems to be a significant enrichment in especially this loci, which we identified as potentially selective for 90-meter fish. So to sum up this part on the temporal variation, we found that introgression during cultural eutrophication affected an entire radiation. So there has been introgression in all the 3 different species, and that the breakdown of reproductive isolation really followed the breakdown of the ecological niche. We also seen that introgression affected various regions all across the genome, various genes have been affected, and we see some pattern which could be consistent with a transfer of potentially adaptive variation from the extinct species into some of the other species, fish which we still see in the lake nowadays. And so to bring it all back together into like my vision for my lab, like we've seen that evolutionary processes like divergent natural selection and gene flow are shaping the genomic variation we see in this whitefish assemblies. We also see that they're heavily affected by anthropogenic environmental changes. But we also see that potentially this like changes which go on and these evolutionary processes go on do something to the evolvability of the species we still see nowadays. And I think also with these identifying some of the genetic architectures behind some of the interesting traits of whitefish, potentially we are getting a better idea of which architectures can facilitate, for example, adaptation or rapid speciation in systems. And with that, I want to say thanks again, especially to Rishi and David, which really like have been fantastic PhD students, which really did all this work and made all these nice figures, which I could present today. Also to many other collaborators on this project and funding agencies, which funded actually this work. And thanks again, Amanda, for giving me the chance to present all this work here today.
Thank you so much for sharing this work. It's been fantastic to see some of the research in preparation that is not yet published. I see some clapping hands, some appreciation from people attending. So I just have a comment to say that, and please, if anyone has specific questions they wanted to ask, you can raise your hand or add that into the chat. But I really think it's fascinating to see how similar the patterns in the genomic architecture between what we see in North American crigonines and the European crigonines is different than what you tend to see in salmonids, in salmonines, because a lot of what we're looking at seems to be very polygenic. So a lot of different areas of the genome seem to be playing into things that we look at, like gill raker size and potentially body size differences. And I wonder, you know, if that underlying architecture that we're dealing with that makes those genome assemblies so challenging is one of the things that leads to the adaptive radiations that we see in these chorogonines. So Bo, I see you have your hand raised.
I don't know if Feulner wants to comment on what you just said, or?
I'm happy. I think I totally agree with you, Amanda. And I also think that's what I thought was exciting to see all this polygenic architecture. And I think there have been like also people like from the pop gen perspective, which have suggested that specifically like polygenic architectures could potentially be advantageous for a rapid adaptation. So in some ways, like it's some things fitting nice together. I don't like, only because things fit nice together, it's not a proof, but it's nice to see that they are fitting nicely together. Yeah.
So I just maybe have a few quick hitters for you, Feulner. I'm not an evolutionary biologist, so I'll mostly stay away from that. But the pattern that you showed in terms of more similarity within a system relative to like an ecomorph in another system, that's one of the things that we're wrestling with in the Great Lakes in terms of how to explain that pattern. And if that could be the pattern, which I don't know that there's 100% agreement on, one of the reasons for that is the potentially relatively young nature of our lakes. So 10,000 years since the last ice age. I mean, is the similar, like how long have those, like has Lake Constance been able to potentially undergo this radiation into the 6 morphs? Is it about that same time? Is it a longer period without disturbance? I mean, sort of before the eutrophication disturbance, I mean, but what sort of evolutionary timescale do you have relative to what we have in the Great Lakes?
Yeah, just like I don't know much about the Great Lakes, but what you said, like all these like Swiss lakes are also post-glacial lakes. So Switzerland has been under a glacier in the last glacial maximum. So all the lakes only formed after the ice retreated. So I think it's a similar time frame. It looks like, so we see 2 mitochondrial lineages in Switzerland. So it seems to be that there have been 2 glacial refugio, and that Switzerland was colonized by 2 different lineages. Nevertheless, mitochondrial lineages like explain no of the species variation. Like there's like one mitochondrial type which is more prevalent. It's most of the time, and there's one which is like rarely found, but there's no association with any of the ecomorphs with the mitochondria. And the nuclear data really suggests that fish within a lake are each other's closest relatives. And we also use some of the D statistics to actually test that. Does like a tree which groups fish by lake instead of ecomorphs explain the data better than the opposite? And that's true. It's always the better explanation based on the data we have. I guess it's difficult to say but I really find the alternative explanation way more unlikely, but it's very difficult to rule out, like that these fish have been colonized by different ecomorphs, and then they have shared so much, there must have been so much gene flow amongst them that they now look genetically like more similar and are only differentiated at those regions which make them phenotypically different. I find, that more unlikely that they have been colonized by, it's difficult to say by what they have been colonized, but by something which maybe has had like a lot of standard genetic variation, which you would expect if 2 lineages came together, that there has been a lot of variation in the initial colonizing lineage. And then they have managed to differentiate very rapidly.
Yeah, very similar discussions from what we have, and I can't speak to it as well as some of our genetic colleagues might. But my second quick question, more from a management perspective, you kind of used the term ecomorphs and species interchangeably, and when you publish the papers, of course, they have a different, C and then a different apparent species name. Are these managed? Are these recognized as species, like from a management perspective, or are they sort of recognized as ecomorphs? That is sort of an interesting sort of subspecific variation that managers may or may not choose to sort of manage.
So I hope I didn't use ecomorphs and species interchangeable. So I really like, they are all described species. So they are like, some of them have not, but I think nowadays, like they are like, there are more and more taxonomic papers coming out of EAWAG, which have described them. And I use the term ecomorph to describe that different distinct species from different lakes, which are not each other's sister species, share an ecological and morphological similarity.
Okay, that's helpful.
That's how I would call, like, what I would use the word ecomorph for. And then within Switzerland, it's unfortunate, like for the fishery statistics, they all call everything whitefish. And they don't report species patches separated by species. While the taxonomic description of the species is very much corresponding to German names for all the different species. So the fisherman, even more, like I'm a genomics person, like sometimes I feel like this fish look very much alike. While like the fisherman, which actually catches fish, they have different names for the different species, and the species concept and their German names are like very well aligned, and they can distinguish them. And for example, it's interesting that, for example, the temporal data on Lake Constance was only possible because there has been this historical scale collections which were collected to determine growth rates. And they are like scales in little paper bags which have been labeled by the species. And like the German name, but the species. So those people which like manage this fish can totally differentiate the different species. But when they report it for the statistics, they report it under the genus name. And so I think they have become more and more aware. And for like, for example, one hatchery I visited, they can differentiate the species, and they try, and they know that they should do it that way, that they should not, that they should breed them according to the different species. But I don't think that has been historically been managed like that all the time.
I have more questions, but I'll wait. Very helpful. Thanks so much for clearing that up about the ecomorph and the species. That makes a lot of sense.
Okay, great.
Well, I'll jump on with some follow-up questions because I'm seeing Bo smile at the same time I'm smiling, because one of the things that's been such a pleasure reading your research has been how similar it is to what we are working on and doing here in the Great Lakes, because we have seen a great drop in diversity of described species from the Great Lakes. And we do have scale archives from these collections. And so that's part of what we have started working on now, is trying to compare these historical collections to contemporary collections in very, very similar ways. So it's really nice to almost have a roadmap from your research to sort of work through some very similar questions we have on our end. And to that point, I just wanted to ask a little bit about, so from the data that you have from Lake Constance, there is some indication of either what we see termed a lot speciation with gene flow, where there's still some active integration at low levels between forms, or maybe it's just a little bit of incomplete lineage sorting remaining in the genome. How difficult is it to tease that base level apart from what occurred sort of after eutrophication? Was that difficult at all to compare where integration was occurring before and after, or was that pretty?
I think in some levels, like having scales from different time points helps a bit, right? Like, so like, for example, that D test, when you have the scale samples, which like are from the same time and the extinct species, and then you have the contemporary samples. If you then identify this excess allele sharing, this to me suggests by the timing also where these samples are from, that integration must have happened before eutrophication and now. So it must have happened during that eutrophication period. I'm still super interested, but we haven't done that at all, like to look a bit more into the length of this introgressed blocks. I think that could potentially also like give you a time perspective of when something has introgressed, but we haven't done that yet. So maybe I've lost it. Like there were multiple parts to you.
There was a comment, and then there was mostly the question of teasing apart the sort of constant low levels of gene flow that might be occurring from a specific time point effect. But I also see that Kimo has his hand raised. Would you like to ask a question?
Yes, thank you. Very, very interesting talk. I would like to go back in history. There was Boo already said about something about the lineages. So you said, if I understand it correctly, that some of the lakes were colonized by two different lineages, at least according to mitochondrial DNA. So do you have any understanding on whether these different lineages colonized the lakes at the same time? Or was it that one lineage were able to colonize them before the other came in?
No, I don't have any knowledge about that, and I don't know if there's any. I can't think of a paper which looked at it. It's still also fairly unclear to me how, like, did these two lineages come together and then the hybrid lineage colonized Switzerland, or was it like you described that, like two different lineages coming? So it seems to be that to my best understanding of the mitochondrial data, it could be that like one mitochondrial lineage is from the east and one could be from the west. That I guess would suggest that they more came like together, but I think it's the same mitochondrial lineages which also both exist in the Scandinavian whitefish. I think, to my best understanding, it's the Scandinavian whitefish have prevalent, the mitochondrial haplotype, which is rare in Switzerland, is the most frequent in Scandinavia. But in the mitochondrial maps I've seen, it looks like that almost everywhere there's a small percentage of the other lineage.
Yes, it's usually that it's two, and then it leads somehow to the next question, that the introgression is very important. We all know that, but is it the recent introgression or is it the ancient introgression that is the most important? What do you think? Like, is it the key actually on the divergence? Like on that, you have that ancient introgression that is then sorted out on the specific condition, or how you see it?
Yeah, I don't know if we know that very well. Like, I think it's maybe also a matter of like what you call most important. I think that the event you're alluding to me, seems to be the relevant one, but it's also the one which is difficult to grasp, because we would need to have the actual lineages, right? The ancient lineages to even tackle that question. So what we did is maybe a bit more the easier. We more looked at like introgression, which is more likely like a later type of introgression, which is like the secondary contact scenarios, which we also which fit with the records of how fish were transported across Switzerland. So it's I think on the grand evolutionary scheme, but there might be a subjective opinion, I think it's the less relevant one. And I think the ancient one, to me, is the more interesting one, but it's the one which I don't really know how to tackle it yet. But yeah.
Yes, thanks. I guess none of us has the answer for that, but it's the hearing of you. Thank you.
So I see Eric Crandall has a question.
Yeah, hey, great talk. I really enjoyed it. I arrived a little bit late, so I missed some of the background. So I apologize if you already answered this or if this is the answer to this is obvious. But it sounded like that these lineages occurred in multiple lakes throughout the Alps, and this lake has cultural eutrophication, but perhaps there are other lakes that are not so impacted. Have you looked at those lakes to see whether the same levels of introgression are happening or not, to try to really isolate whether this is due to eutrophication?
Mm-hmm. We didn't with the whole genome data, but the actual study, which like I have the overview of, where we maybe you missed that, where we've seen like how many species of the historical described species we lost. There's a correlation with how much, or like, I think it's the maximal phosphate during the eutrophication period, which was measured. There's a correlation between that and how many species got lost. And based on microsatellite data in that von Landen paper from 2012, they showed that the decline or like of genetic differentiation, or the differentiation between species within a lake, is more pronounced in the lakes which were less affected by eutrophication, and has the differentiation between species in the lakes which have more been affected by eutrophication. So they are less affected there. So there seems to be this correlative evidence, but that's all in the 2012 paper and based on microsatellite data for the genetics part. Yeah, so I think it fits everything like we've seen. Like I did one study using rat data on two lakes which are like used to be one lake, like Lake Zurich and Lake Wahlen, and so they have the same two species that has been historically one lake. So they are like and there, but Lake Zurich, which is closer to the capital of or to a large city of Switzerland, has been more affected by eutrophication. And there you see that these two species are less differentiated. And then Lake Wahlen, which used to be like less affected by eutrophication, the differentiation between the species is larger. So it all fits in the direction you indicated. But yeah.
Cool, thanks.
So I have another follow-up question in regards to the integration, which is that's something we're still testing in the Great Lakes, but one of the things that has been well documented is convergence of traits, morphological traits from different, more distinct spaces historically to something intermediary. And that's been used to hypothesize that integration has occurred, but we haven't had the opportunity to test that yet. And that's actually some of the work that we're doing. And I was curious to see if you will see very similar things happen in the whitefishes in those alpine lakes, where with increased integration, you see convergence of like gill raker counts or other like sizes or other things like that that track very well, or if it's something that's a little bit more stochastic in nature.
Yeah, again, this has been already neatly shown in that von Landen paper. In the von Landen paper, I think they looked at the historical gill raker counts for those species, and then they counted the Gilreka counts of the fish nowadays. And again, there's like exactly that, like what you're describing, and again there's a correlation with the severity of the eutrophication, having merged things more. One thing we're currently looking at is, of course, we don't have the phenotypic data for the samples of the scales. But the neat thing is we now have that one locus which we know is correlated with Gilreka count. And you can see that the allele frequency differences at this locus in the old scale samples has been more. And it like also goes in that direction, that they are like the frequencies become more similar over time. Of course, that's just like, yeah, one locus is like, we know that locus doesn't explain everything, but like it's interesting to see. Yes, it's again very, very similar to what you described.
Bo, you have a question?
When you were talking about reproductive isolation, Philine, you talked about sort of by depth mostly, going down to 90 meters. In the Great Lakes, we see reproductive isolation among our Coregonine community as both depth and time. I mean, how well studied is it in terms of the reproductive life history of all the different species in a place like, I think Lake Constance, was that the one with the most species? Was that the most diverse, or?
No, actually Lake Constance nowadays has 3 species, used to have 4. Here in Lake Luzerne, we have 6 different species.
Okay, so like the one with 6, like, is it all space, or is there some temporal component too, or is it not really well known?
I think there's definitely also a temporal component, and I think it's both. So for example, the fishermen do spawning fisheries here, and even like many spawn in winter, right before Christmas. But they are also, I think they know that certain species are a bit earlier and other species are a bit later. And especially in the species which spawn deep, there are like descriptions that there has been summer and winter spawners.
Well, summer too, huh?
Yeah. It's only like the deep ones. I think it's only the deep areas are cold enough for them to spawn in summer. But and that like I don't really like, I think that's not always entirely clear to me to which respect summer and winter spawners are different types of the same species, or to which respect they're like have been, I think potentially then we lost many of those summer spawners, because I think there are more historical descriptions of those, and but it's also not entirely clear to, like, while we know a lot of them because the fishermen are interested in them, they are nevertheless, they're also like set in their routine, right? Like they are now doing spawning fisheries in winter. So it's like, I'm not sure to which respect someone really looked at. I think I think they know there are like still a few fish which are ripe in summer as well. But
yeah, we have kind of the same thing where it's fall spawners all the way into winter, spring, and then even early summer. We have some evidence historically of running up in tributaries and then all the way into the deepest parts of the lake. So all of that habitat heterogeneity probably was really important in helping that adaptive radiation. And I was just thinking about that reverse speciation and what, like behaviorally, or what was it that caused those deep water forms, you know, as the lakes got less productive, I guess just not being able to reproduce in the deeper waters, moving into the shallower waters. And if there wasn't as much time separation as space separation, that really would lend more credence to the, you know, to the explanation. So
what I sort of like, when there was the eutrophication of Lake Constance, we know that like we have historical records of oxygen levels in the deeper sediment areas where this fish eggs would have needed to evolve. And those oxygen levels, to the best of my knowledge, were too low for those fish embryos to develop. So what I think makes sense to me is like that the ones which spawned deep didn't develop, so they were not successful. Only those which were like sort of overlapping in their spawning grounds with other species sort of still reproduced. And under natural condition, I think those these little bits of gene flow would maybe not like have had an effect, but like suddenly those were the only ones which like survived. And then like that's what we are like nowadays seeing. I think the other interesting thing is that the productivity of the lake shifted. Like there was suddenly way more pelagic food available. And I think beforehand there must have been this strong divergent natural selection, like favoring either the one or the other type. But if then productivity of the lake is just increasing and like there's plenty of food and it doesn't maybe matter like how well you're like capable of getting it, then like also that like hybrids don't have a disadvantage anymore, right? Like it doesn't matter, like you're surviving because there's plenty of food.
Yeah.
So I think the combination of both things happened, but like that's just what I think and what makes sense to me.
Yeah. Thank you.
Kimmo, you have a question?
Yes, thanks. Very interesting questions. So just on, are they all lake spawners or do you have any river spawning populations?
Historically, there might have been more river spawning populations. There's still like a population which nowadays seem to spawn in a channel between Lake Zurich and Lake Wahlen. There used to be a natural connection of these two lakes, and nowadays they are only connected via a channel, and we can catch spawning fish, or occasionally fishermen catch spawning fish in the channel. So that it could be that Lake Constance has another species which spawns in the rivers. But the rivers in Switzerland are also like not that easily accessible. Like there are like lots of power plants and lots of human-made barriers nowadays. So I think we lost a lot. I think historically there must have been more fish which migrated and spawned in rivers, and I think they're now rare or gone to some extent.
Okay, so mostly lake spawning populations. So what is your view on how this spawning time actually diverges? Like on how it is possible that we have a summer and winter spawning population and how that initially diverges? How it happened?
I have no idea. Sorry, do you have an idea? Did you? Like,
yeah, you know the physics of the lake, so you know it's, as you mentioned, that it's, if you are spawning on the deep, so it's kind of also on the amount of daily decrease and so forth. So is it still that all the mobs like hatch at the same time, or do you see like a difference on the hatching times?
So that I don't know. I don't know if someone looked like at the natural hatching times. I think they have their hatching times from the hatcheries, but they are given by the temperature they are like raising the fish in. To the best of my knowledge, really, like we only have summer spawning or indications that they're summer spawners for species which spawn deeper, like as you said, because I think that's everything I know. I don't know if someone investigated that more in depth.
Yes, so it's usually the hatching time is very, very important. Like I think Louis Bernascez and these guys look on this, that it's very important that you hatch on the correct time just to match on the prey resources. So, but on the habitats has a bit different temperature, like the dynamics and so forth. So perhaps they still hatch around the same time, maybe a slight difference on that. But yeah, once again, a question that it's not very easy to answer. But very crucial on the reproductive isolation. So thank you.
Yeah.
So we've had a couple of good questions come into the chat. Sarah Salisbury asks a follow-up question about the lineages. Do you think the Lake Constance radiation represents an example of combinatorial speciation? Or do you think that the radiation might have been possible without the genomic input of both of those lineages?
I think, yes, I would think of this like something like this combinatorial view. And because the two lineages, like we know that they exist, I think they very likely played a role. I don't know how you can test if without that signal. I think exactly that happened, but yeah, it makes sense to me.
If anybody comes up, I was going to say, if anybody can come up with a good idea of how ways to test that, we have a system in the Great Lakes as well with overlapping mitochondrial haplotypes that don't necessarily seem to be very clearly geographically distributed. So if you have anybody coming up with a way to test that, we could add another system to look for more into radiations and the associations of overlapping lineages and that sort of combinatorial speciation. Mm-hmm. So Sarah commented again, fair enough, thank you. So there is another question from Scott Hansen that says, is there an understanding of whether thermal stressors have also been implicated in whitefish recolonizing the deeper areas of these lakes? Or is the feeling that it's mostly forage and reduced eutrophication related?
I don't know that. I think actually these lakes are like super well studied and I just don't know all the details. I'm pretty sure people know how this thermal habitat has changed over time. I don't know if anyone looked at like to which respect that is explaining some of the things we are seeing. I think the eutrophication signal is so obvious and so strong that maybe we all like tend to focus on that, but I think that's I think it would be really good to look into that more, but I don't know if anyone did.
All right. So I have a bit of a methodological question for you, which is you were able to get nice genotypes from some of your historic samples. Can you give us, those of us that are getting into doing more whole genome work on scale samples that are, 100 years or very near 100 years old, how many attempts it took to get the samples that worked for you?
Yeah, so now David did like lots of his work is done during his PhD, but he started that as his master's. And when he came to me and said like, I want to do that, I was like, are you sure you want to do that for your master's? You could be like extracting a lot of samples and be super frustrated and have no results. And he totally proved me wrong. In our case, we were very lucky. I think it's like I think two things are critical, or like where we've been lucky with. Like it's really like that it is this collection. So there are multiple scales in bags. So we're not extracting DNA from only one scale. Like I think he used something between 4 and 5 scales per extraction. And it's really like what he also could show, we have like, it's a big collection. And there are certain batches which work very poorly and other batches which work like this. And what I think so, and they're like very like, so it's essential, like the DNA actually comes from the tissue which is on the scales and not from the scales. So if someone's cleaned the scales, it's useless, right? So it's really like the leftover material. I think it's a bit like, definitely like the storing in paper was essential. So if they're in these paperbacks, I think we have a few with plastic, which like didn't work. I think maybe it's like how dry they were stored or how cold it was when they were taken, which might influence it. So we definitely see this batch effect. But in the good batches, like he takes 4 or 5 scales, does the extraction, can measure DNA, the negative control is negative. So it's like, and the data is sort of, the DNA we extract is more fragmented than, of course, than the contemporary one. So to some extent, like the libraries are a bit different. Sometimes we have overlapping reads, so we have to merge the reads. So they're like the read length is a bit smaller. But we didn't observe any, for example, DNA damage at the end of the reads. So there's still like pretty good DNA. And they're like now, I think the last batch we sequenced, they started to develop more and more of these kits for like, which are for this more ancient type of DNA. And then the library prep also works really nicely. And it's like really nice data you can get.
Well, that's fantastic to know. I might have to reach out to you guys to see which kits you're using.
Yeah. We actually like, I think we just use a quietin kit, and I like it's like I think he uses a different buffer for the extraction, but it's also all in the paper. But like, yes, please like drop me an email and like we're happy. But I really like don't think it's like it's everything is in the paper. We don't do special things.
Oh, it sounds very much like your experience has been with ours. I think it's one of the fun, exciting things for fish biologists to really tap into these days, because for a long time, people were aging with these scales and looking at growth with these scales, and then they decided, okay, this isn't the best way to look at it. So they just sat in closets or sheds, sometimes outside. A lot of collections that we have records of, we think just got tossed because people weren't aware of their value to us at these times, being able to get DNA off of them. So I would say our experience tracks very similarly with yours, where it's very patchy.
Yeah.
Nate, back in Stos, you have a question?
Hi, just a follow-up to Amanda's question there. I was just wondering, when you were analyzing these scale samples, if you have to take any approaches that would account for DNA degradation, like a genotype likelihood, or you could make hard SNP calls from the scale samples.
Yeah, so they like there's no damage signal at the end of the reads. We looked into that. The data is a bit like, because the reads are a lot shorter, we usually have a bit less coverage for the ancient samples. It has been a bit of a challenge to bring these data sets together, and like you need to like play around a bit, like, or you have to be careful with the filtering. Ideally, like we run a lot of things just on the genotype likelihoods, and preferably most of the analysis just directly in angst on the genotype likelihoods. But for some of the data, we at least feel like the data set is good enough to, if we need to, also call genotypes. But it's mainly because some analysis you can't do on the genotype likelihoods.
Great, thank you very much. Wonderful talk.
Thanks.
All right, do we have any other lingering questions or comments out there? Looks like we've reached the end of immediate questions. Well, thank you so much for coming and talking to us about your alpine whitefish research. I think it's really exciting how well the systems often track with each other in terms of the radiations that we see and the signals that we see. And I think that there's a lot of exciting potential research to try and combine the patterns that we see in these, oddball chorogonines in terms of salmonids to try and better understand them. So I really appreciate you coming and talking with us today.
Yeah, thanks so much. I really enjoyed it. And thanks so much again for inviting me. It has been a great pleasure. Thanks.
All right.